Matthew Fuchsia. He's going to talk about acquisition of thiamin by wild Lake Trout eggs and 3 embryos reared in Lake Champlain.
Yeah, so unfortunately Colin couldn't be here today. If you know Colin, you'll probably quickly recognize that I'm not like him, much smaller. But I'm happy to give this talk for him today. I'll try not to stumble over it too much, but helped along the way with the project, which is a major part of his master's thesis at Stony Rockport, looking at the thiamin acquisition of lake trout and trying to look at some ways that these fish might be able to mitigate the effects of thiamin deficiency on their own. So thiamin is this essential vitamin, which means that it must be obtained through the diet and has a ton of different roles that are really important for fish development and just fish survival in general. It's this cofactor for cellular metabolism, so it's really important for producing energy. It has a lot of functions with neurological development, and then it can also serve as an antioxidant. So when you don't have enough of this vitamin, bad things happen. And this typically occurs at early life stages where you see a lot of mortality for juvenile fish, some point between hatching and the swim-up stage. And the picture here is showing some lake trout that are all still alive, but they're just suffering from this thiamin deficiency. And you can imagine if this is a fish out in the wild, while they might be alive here, that's going to look like a great snack to any predator, and you're probably not going to make it too long, even if you don't get consumed by something else. So a lot of the symptoms include abnormal swimming, lethargy, there's some issues with the immune functioning, and then also, ultimately, if you do have severe enough, or if your concentrations are significantly low enough, it can lead to mortality. And because of this deficiency has been identified as a potential cause of limited lake trout recruitment in the Great Lakes region and across the range where lake trout overlap with alewife, which I'll get into it in a little bit, because that species the alewife is associated with causing this thiamin deficiency. And there's two different hypothesized mechanisms that cause this deficiency. One's related to consumption of high lipid contents. So if you eat a lot of lipids, you have a greater chance of lipid peroxidation or oxidative stress. As I mentioned on a previous slide, thiamin can function as an antioxidant, which then counters that oxidative stress. You might have depletion of thiamin if it's being used for this purpose. But then also thiaminase consumption. So thiaminase is an enzyme that can break down the thiamine, and as a result, you have less available. So as I said, alewife are known to cause thiamin deficiency, especially when you have a ton of alewife in your diet. There's some great studies back in the early 2000s that had a controlled lab setting where they fed lake trout as adults alewife for up to 2 years at different amounts, and they found that if you have just as little as 35% alewife in your diet, you'll have reductions in your thiamin concentrations of the eggs of those adults after feeding for 2 years. And this results, as I said, from increased thiaminase activity and then also a high lipid content of the prey. And the figure here is just showing a comparison of lipid content of 3 different common prey species found throughout the region. And this is during fall, but it's consistent throughout all seasons. And again, you can just see that the alewife have much higher lipid content. But this relationship between alewife populations and lake trout, and really salmonids in general, is somewhat complicated. The general consensus is that when you have alewife and lake trout coexisting, bad things are going to happen for the lake trout. If you remove those alewife, the lake trout are going to rebound, and that's what the figure here on the right is showing. This is a paper from Lake Huron, and the red vertical line demonstrates the point at which the alewife population in Lake Huron collapsed, and then you can see quickly after that the thiamin concentrations rebound, and what also happens is you see this increase in the recruitment of lake trout in that system. However, in Keuka Lake, as you might have heard from a talk yesterday, alewife and lake trout were able to coexist until the alewife collapsed. But they were still coexisting for a while. So we have lake trout that are doing well when alewife are around. And then more recently, in Lake Champlain, you have lake trout that are being stocked for a number of years. Alewife are introduced, and after that introduction, you now start to see recruitment of the lake trout, almost seeming like the alewife helped the lake trout come back. So there's a lot of confusion going on here, uncertainty, and so we're trying to figure out some ways that this uncertainty can be explained. And we know that thiamin deficiency can be mitigated in hatcheries through different treatments. So you can treat the eggs early on in a water hardening bath for only 1 hour at these high concentrations, and then that'll provide enough thiamin to the eggs to allow the offspring to survive. You can also treat the fry after hatching. You need a little bit of a higher concentration, but you can still mitigate the effects of thiamin deficiency later at a later life stage and see great responses. And it's a really rapid response. You treat these fish, and by the end of the day, they're looking much better. You might remember that figure I was showing, or the picture I was showing before with all those lake trout on the bottom. There's a good chance that if you add thiamin to that water, they're going to start swimming around by the end of the day. And then lastly, you can also do injections into the adults with their body cavities. Now, it's not going to help the adults so much, but if you do this a few months prior to spawning, you can increase the thiamin concentrations in those eggs, and that way you have offspring that are able to survive without having to do any treatments at the juvenile stages. So we know that if you have a lot of thiamin in the ambient conditions, these eggs and offspring are able to acquire that and recover. So we did a pilot study where we looked at the thiamin concentrations in the water just to see if they are out there. There's very little literature on this, particularly for the freshwater systems. And what we found was, yeah, there really is some water, or some of this vitamin in the water, and it's pretty variable, but we looked at some municipal water sources. So at Stony, Brook, and the University of Vermont, we took water samples from our lab, sent that out to be analyzed, and then we took samples from pelagic water in Lake Ontario and Lake Champlain, and then also a bathytic sample on Lake Champlain, just to see how things might look. And really what we found was that there's a lot of variability. I'll get at the method for this in a little bit, but it's very complex, so this variability is largely driven by us having a few samples. I think there were maybe 2 or 3 samples for each of these averages. So I think it was 3 samples for each of the averages, but it's hard to do. But we at least know that there is some of this vitamin in the water. And there was also a study that was done probably about close to maybe 10 years ago, almost, but where there were lake trout eggs that were taken from Hugo Lake, which is known to have thiamin deficiency, and the eggs were fertilized, and siblings were put in a hatchery system at Stony, Brook, and then some other siblings were put in a hatchery system at the University of Vermont. At Brockport, they saw signs of TDC and high mortality, whereas siblings raised just in a different condition at the University of Vermont had no mortality and no signs of TDC. So this was a real head scratcher. And another thing that was noticed, just for the eggs at the University of Vermont, because they weren't looked at Brockport, the concentrations of thiamin increased during development. So this was kind of like, what the heck is going on? Like, we're not adding anything to this water. There must be something going wrong with machines. All the eggs, all the thiamin concentrations, they were all run at Stony, Brockport, so it was all done with the same instrumentation, same methods. So this was a real head scratcher. Another thing that's important, at least in wild settings, is that there's a lot of thiamin-rich prey out there, and wild, what we're calling free embryos, they're the lake trout after hatching, they still have that yolk sac. They're going to have the ability to feed on those zooplankton that are really rich in thiamine, and they can start feeding in less than 2 weeks after hatching. So that's, if they're able to swim around and they're able to acquire prey, they might further increase their thiamine concentrations just by feeding on the food that's available to them. So this project really had 2 objectives, looking at if these offspring are able to alleviate thiamine deficiency on their own. First one, if the thiamine concentrations in lake trout eggs are influenced by water thiamine throughout incubation. So from the time that these eggs are deposited by the female, all the way throughout the period until they hatch. And then also comparing thiamine concentrations between, between the hatchery-reared embryos and those in the wild. So if we have fish that are raised in the hatchery setting, and we have siblings that are raised in the wild, are there any differences in their thiamine concentrations, just based on the resources that are available to them? So I'm just going to warn you, before I go to this next slide, it's very complex about the experimental design. I hope it's not too overwhelming. I'll try to walk through it slowly. But here's essentially what we did. We have 20 lake trout that we'll start with. We're going to go from the left part of the slide all the way to the right. And the thing to keep in mind here is that this is an experiment where we're splitting siblings between a hatchery setting and a natural setting. So we have a bunch of eggs that are collected from this lake trout, and we take a bunch at that point, just right from the female. What is our baseline thiamine concentration? We then split those eggs into another batch of eggs into two different groups. One that's fertilized on reef water, and another one that's fertilized on this dechlorinated city water. And so we keep these eggs separated at all times, and we're completely operating with the water that that egg is going to, or those of the offspring are going to be reared in, whether it's natural water in the gray, or if it's the city water or municipal water that has the white background. After fertilization, the eggs go through, some of the eggs went through a thiamin treatment during the water hardening. After water hardening, we do another egg sampling event to see if there's any increase in thiamin concentrations just from that water hardening process. These eggs are really swelling as they're taking in water. They're also taking in a lot of milk, so there's potential for these eggs at this early stage to already be taking in a lot of thiamin. After that point, on the reef, these eggs are put into 3 different bags. You can see an example of that in this bottom left picture, where we're depositing eggs into these controlled chambers on the reef that allow the water and some invertebrates to pass through, and they sit there throughout the winter while the eggs are incubating, and then they also allow the fry to stay inside, so after the eggs hatch, everything is as natural as can be, and we have this great comparison for the lab setting where they're just raised in these hatching trays, put into tanks, and resembling more of a hatchery setting if you don't have any thiamin supplements. So we've got siblings raised in these 2 different conditions, and we do treatments at different times. So we have a first treatment, our first collection, where it's right at hatching, or ideally right at hatching. It's kind of hard to say when these are all out on the water, but collecting eggs and free embryos in the wild, and then also in the lab, and then also 5 weeks afterwards to see if there's any additional increase in thiamin concentrations by potential feeding, as well as just accumulation. The thiamin can get acquired through the gills of the free embryos. So there's a lot of different ways that we're thinking these offspring might be able to get this vitamin. And we were also looking at the dissolved thiamin concentrations in the water for each of these different periods. And there's just a quick cartoon showing what this might look like. So we start with our water collection, we filter it, we then do an extraction of the vitamin, and then it gets quantified in the liquid chromatograph and then a tandem mass spec. And we looked at not just thiamin, but there's thiamin precursors, and then also degradation products that give some insight to how this vitamin is being used. And as far as the thiamin concentration in the eggs and free embryos themselves, everything was done in duplicate, so we would have 2 different groups from each of those, like the bags, or from each of those little, the cups that had the eggs, for example. So for each family there was a duplicate, and then this was quantified using the high performance liquid chromatography, which is shown on this side. And then this is just an example of the output that you get with different concentrations showing the amount of the vitamin. And we also freeze-dried these samples prior to doing the analysis because, like I said, when these eggs are going through water hardening, they're taking in a lot of water, so that can cause some dilution effects. By removing all that water, we're just looking at the thiamin concentrations and how that changes over time. So this slide is going to be going through some of the thiamin concentrations in the water itself. These data are somewhat new to me. I haven't had a lot of time to look at them, and in general, we haven't had a lot of time to look at them. And as I also mentioned, looking at dissolved thiamin concentrations in water isn't really well documented. So there's a lot of stuff here where we're seeing this really high variability, and we're kind of, well, what does that mean? We don't really know yet, but it's something that we're hoping to look into. And so starting with thiamin here, like I said, we just see this really high variability, but we're looking at thiamin concentrations across time and based on the 2 different conditions, whether we're in the field or in the lab. But we're seeing that there is thiamin present, which is the key point here. There is thiamin available to these offspring in the wild. Next, we're looking at the thiamin precursors and the degradation products, and you're seeing that these values are really all over the place. And again, it's right now we don't really know how to interpret this, but it means that there's likely stuff that's going on producing these precursors that may then be available to the offspring, and then also the degradation products might be that they're being used by these offspring and then put back into the ecosystem. The more exciting results that I'm looking forward to sharing with you all right now is the change in the thiamin concentrations over time between the hatchery and the wild setting. So starting off here, you'll see we have the dry thiamin concentration on the Y there. Purple being the field, green being the lab setting. Starting off with the unfertilized eggs, we've got 10 families that survived throughout the entire period for the wild fish in the natural setting, and then we had 18 families that survived throughout the entire period in the lab setting. So there's 4 different time periods that we're going to go through, and for each of these time periods, it's going to be samples from those same families. So at the start, it makes sense. These are unfertilized eggs taken right from the female, and we're seeing that they're very similar. That makes sense. After fertilization, they kind of drop down a little bit. So maybe there's some stuff, there's a lot of energy that's being needed for that fertilization. Maybe there's something going on there. Now here's where things start to get interesting. At hatching, and this is for the fry, we're already seeing a jump in the thiamin concentrations for those wild fish. 5 weeks after hatching, that difference gets even greater. That's a huge jump in thiamin concentrations for these fish in the wild. Is this attributed to feeding and the water, the thiamin in the water? Probably, but it needs a lot more work, and that's something that we're looking forward to studying as the study continues to move forward. So, takeaways from this: dissolved thiamin concentrations is highly variable in both lab settings and in natural settings. And we see these thiamin precursors and degradation products that have high concentrations and potentially indicate that there's this high turnover of thiamin, this high use of thiamin by all sorts of organisms. We also see that lake trout eggs and free embryos can acquire thiamin during development, and this has a lot of implications for what does TDC, this thiamin deficiency, really mean in the wild. Is this really as much of an impact as we're expecting it to be? It makes sense that at least alloy has some sort of impact on lake trout recruitment based on what we're seeing in these other systems where lake trout the alloy collapse and the lake trout recruitment increases. But there's a lot of reasons why alloy might contribute to lake trout or limited lake trout recruitment. They're also known to feed on the fry, which might be causing that limited recruitment. And then also, as I mentioned, we made some modifications to this experiment. Here's Colin again, showing the new rearing device that we're using. So we've got cinder blocks. We got some issues with escapement, and we're also looking at ways of keeping the free embryos, so we can look at stomach contents of these, see if they're actually feeding. Is there a difference in the feeding? I mentioned earlier that we did some treatments in the study. The treatment wasn't that